Thank you, Lisa, for the nice introduction. Yes, I'm Alice Berger out at Fred Hutchinson Cancer Research Center in Seattle, Washington, and I have no relevant financial relationships to disclose. Now, some of you, if you've been to Seattle, may realize that this inset photo here is not Seattle, Washington. In fact, that's Morgantown, West Virginia, my hometown where I grew up for many years. Morgantown is a beautiful, idyllic college town a few hours south of Pittsburgh, Pennsylvania, with a population of 31,000. And the reason I'm telling you this is because that was the context by which I was shocked to find out that cancer causes many, many, many more deaths per year than the population of Morgantown, West Virginia. And in particular, lung cancer is the leading cause of cancer deaths worldwide and also in the United States, and also in both men and women, which I think many people still don't know. In fact, together between men and women, lung cancer kills over 135,000 people every year in the United States, which is over 4 times the population of Morgantown, West Virginia. And so as I started studying cancer biology and learned about this, that was part of my motivating reason to study this disease. Now, there are many risk factors for all types of cancers, and these risk factors can either be intrinsic, such as genetic factors, or extrinsic, such as environmental factors. And on the intrinsic side, you can have, for example, genetic mutations. Kind of well-known one is BRCA1, which causes hereditary risk for breast and ovarian cancer. On the extrinsic side, of course, for lung cancer, one of the the highest risk factor is smoking exposure. However, as our understanding of lung cancer has grown in the past decade, so has our understanding of the underlying causes of lung cancer. Back in the 80s, when smoking was incredibly prevalent and we had seen this really high increase in lung cancer cases, there was a massive anti-smoking campaign that was very successful from the American Lung Association and the government and others. And you can see that reflected back in this button from 1986 that said, "Kiss your butt goodbye." Now, what happened is that was remarkably successful, and we've decreased the incidence of lung cancer over that time. However, what we see is there's still a large population of people that are getting lung cancer that have never smoked. And we were kind of blind to that before when everyone was smoking. And you can see that reflected now in the changing messaging from public awareness campaigns, culminating in 2019 with the Go-To Foundation for Lung Cancer, reminding everyone that anyone with lungs can get lung cancer. And in fact, what is worrisome is that we're seeing an increasing proportion of lung cancer in never smokers. And for the rest of my talk, I'll be talking a lot about never smokers. You should know that in the research community, a never smoker is defined as a person who has smoked fewer than 100 cigarettes in their lifetime. Anyone that smoked more than that would be either a current or a former smoker, depending on their current smoking habits. So in this nice study in 2019, Pelisoff et al. Performed an analysis of 3 different large medical institutions and looking at the percentage of lung cancer patients, either non-small cell lung cancer or small cell lung cancer, NSCLC or SCLC, that were in never smokers. And while small cell lung cancer usually occurs in smokers only and has remained relatively steady, what we see is that non-small cell lung cancer, that is the most predominant type in non-smokers, has increased over the past several decades. And there's a really kind of hot debate in the research community now whether this really reflects a growing incidence of the disease, or if this just reflects the changing smoking habits, and this is a larger proportion of cases in non-smokers. But several studies, including this one, concluded that it does appear that there's an increasing incidence of lung cancer in never smokers for unknown reasons. Now, another interesting fact is that even if you subdivided this to its own disease, lung cancer in never smokers, shown here with the NS, would actually constitute the 7th leading cause of cancer deaths. So it's clear that even if we were completely preventing lung cancer due to smoking, we would still have a significant proportion of cancer mortality occurring due to lung cancer. Another worrisome pattern is that we see that women happen to be disproportionately affected by lung cancer cases in never smokers. So the percentage of lung cancers in never smokers that are in men versus women is much higher for women. So in the United States, about 3/4 of lung cancers that occur in never smokers are in women, and the remaining quarter in men. And we see that pattern repeated regardless of the geographic location, with the majority of never smokers with lung cancer being women. And right now, there are a lot of theories about why this might be, but in fact, we really don't know why women tend to get this form of the disease more than men. Some initial clues came in this study by the Cancer Genome Atlas in 2014. We and others performed mutational analysis in 230 non-small cell lung cancers that included mostly smokers, but also some light or non-smokers. And please ignore the colors for now, that's not important. But what these are different genes that are frequently mutated in lung cancer. And when you see this little symbol, that indicates that this gene was more frequently mutated in smokers, or if it's on this side, it was more frequently mutated in light and non-smokers. Likewise, we saw that there were gender differences where men had certain genes that were more frequently mutated than they were in women, and vice versa. And so it's clear that these demographic factors of smoking status and gender actually influence the biology of lung cancers. And so that led us to think about a few questions. The first was, are there new mutations in lung cancers from never smokers that could be targeted with cancer therapies? And that's one particular angle that I won't have time to talk about today, but we're very excited about. Most of the previous genome-wide studies have focused on smokers, and so we just don't really have enough data on never smokers to have identified some of these potential drug targets. But what I want to talk about today is whether the mutational profile of the cancers themselves can tell us something about the intrinsic or extrinsic genetic risk factors that I mentioned. And so for the rest of my talk, I hope to define to you what a mutational signature is, and show you how smoking actually alters mutational signatures, leading to these molecular differences between lung cancers in smokers and non-smokers. So the challenge to answering these questions has been that the majority of lung cancers sequenced to date are from smokers. And so we simply didn't have the type of genome-wide data that we would need to be able to answer these questions. That's why I was really excited when I joined the Hutch a few years back, and I was introduced to Dr. Garnet Anderson. She is a PI of the Women's Health Initiative, which was a large observational study and clinical trial that began in the 1990s to profile the risk factors leading to breast cancer. And what she and others did was really remarkable, because they followed a group of over 100,000 women starting in the mid-90s, and they tracked their health status over time every year through surveys and follow-ups, and they collected multiple different samples from them, from their normal tissue, and if they developed cancer, they went back and also collected tumor tissue from these women. So talking with Dr. Anderson, we realized there was a really important opportunity to identify a cohort of women who developed lung cancer who had never smoked. And in particular, this is a little bit unique compared to some other recent studies that focused on young individuals. And here we're focusing exclusively on postmenopausal women who have never smoked. As I mentioned, they collected this extensive metadata, and that included not only their own smoking status, but also the history of secondhand smoke exposure, occupational exposures, and other demographic data. And Dr. Sita Priyamurthy is a postdoctoral fellow in my lab, a really talented, wonderful scientist who has been leading the computational analysis of this sequencing data. So the study design was that we took peripheral blood, fresh frozen, as a normal tissue control. And we were able to obtain lung tumor tissue, which is formalin-fixed paraffin-embedded tissue, from each of these individuals. We were able to collect these samples from 57 never or light smokers and 18 heavy smokers that we included as a type of control and comparison group. And then we performed custom whole exome sequencing data, which sequences all the protein coding regions of the genome. We were then able to identify single nucleotide variants, or SNVs, and insertion deletion mutations, or indels, as well as copy number variants, or CNVs. And using this data, then, we can identify previously identified important mutations in lung cancer, as well as novel mutations, which have important implications for personalized therapeutics. We can also perform the mutational signature analysis that I'll show you today, and then link this to the WHI curated metadata to provide insights into the molecular mechanisms of lung cancer development in these individuals and identify potential risk factors that we hope will lead to improved screening and risk mitigation in the future. So before I show you some of the genetic data, I do want to distinguish between germline and somatic mutations. And all of the analysis I'll show you today is based on somatic mutation detection. When we talk about germline mutations, these are mutations that are either inherited or are de novo mutations that occur in the egg or the sperm, so that the mutation is present in the gamete at the time of conception. That means that the variant would be present throughout the entire person's body, in the development, as well as the adult tissue, and in every cell of the body. In contrast, somatic mutations are mutations that occur at any time during or after development, so after conception. These would be restricted to a particular cell or part of the body. And in the case of cancer, we think of them as kind of a tumor-specific mutation, right? So that mutation was present in the cell that founded the tumor, and that mutation is going to be unique from something found in a normal tissue. So both of these types of variants can be important for cancer biology. BRCA1 hereditary mutations are an example of an important germline mutation that confers a risk. But somatic mutations are the ones that might be induced by environmental factors due to exposures. And so those are the ones that we'll focus on today. So what Sita did is she was able to compare the tumor and normal tissue to identify somatic mutations. And then the first thing she did was just assess the overall mutational rate in smokers and non-smokers. And so here she's color-coded the samples from heavy smokers in red and never and light smokers in blue. And you can see that on average, the heavy smokers have a higher mutational burden across the protein coding regions of the genome compared to the never and light smokers, which are in blue. Now, we do see a few outliers of never smokers that have high mutational burdens and heavy smokers that have low mutational burdens. And she's actively trying to dig into that metadata now to see if there are any other factors that could explain these outliers. Now we wanted to look and ask, what are those mutations? And for that, we started to do this mutational signature analysis. And the way I like to think about mutational signatures comes back to this analogy I always like to talk about, but is getting increasingly dated, which is this idea of finding a typo in the phone book. So hopefully some of you are as old as I am, so you remember that there used to be this big, gigantic book where you had to look up people's phone numbers if you wanted to call them. And looking for mutations in the genome is a little bit like looking for a typo in that big book. Before computers, it would have been very difficult, but now with computational power, it's trivial to compare two copies of the phone book and say, what's the difference? And if you do that, comparing the tumor to the normal tissue, you can find in some cases these differences, which are somatic mutations. So let's say we found one here. What that actually looks like in terms of the human genome is you would have a letter or a base that was changed from the normal sequence. So in reality, the human genome is huge, so it's really more like finding a typo in 200 copies of the phone book, but same idea. So then what exactly is a mutational signature? Well, the way I like to think about it is if one of these mutations is in a single gene, let's say a single line of the phone book, then when you think about this mutational rate and all of the mutations throughout the genome, you see there are many scattered throughout the genome, and you might think that they seem pretty random and pretty unrelated. But what if I told you that each of these typos was actually the same change from a C to an A? Would that tell you something about the possible mechanism of how these typos were created? In this case, I would think maybe the printer had a problem that had systematically changed all the C's to the A, and that would tell me something about why these typos were there. So it's the same idea in the human genome for mutational signatures. When we see these patterns of the same type of mutation, it can give us clues about the cause of these mutations. And in fact, C to A mutations are uniquely caused by the carcinogens in cigarette smoke. So if we saw a different pattern, we might think it was due to aging or radon exposure, or in some cases something else. Maybe we don't know what the underlying cause is, but we can see that the mutations come from the same mutational process. So with that idea in mind, CETA performed mutational signature analysis. There have been a compendium of over 30 different types of signatures defined by the Sanger Institute in the UK. And so we use those as reference signatures. They just have numbers here and as an example. And what we see is that smokers and non-smokers have strikingly different mutational signatures. So smokers have an abundance of this signature 4, which is the C to A type mutational signature caused by cigarette smoke. And this has been nicely demonstrated in cells in a dish before, that the cigarette smoke actually can directly induce those types of mutations. In contrast, the never smokers, we don't see evidence for signature 4 mutations. These are just 4 examples here. Instead, we see that almost all of the mutations in the tumor have a pattern of this signature 1, which is associated with aging. So scientists consider this a kind of molecular clock signature, that basically the older you get, the more cell replications your cells have gone through. Cells tend to accumulate these type of signature 1 mutations. So we're seeing very different patterns between never smokers and smokers. CETA is now looking, in some cases we see unknown signatures, and so she's trying to understand if those are really validated and what potential mechanisms might explain that. We know you can get mutational signatures due to intrinsic genetic issues as well. So there are DNA repair defects and other kind of endogenous processes that can give rise to specific signatures. And so she's currently trying to identify if any of those might be at play in never smokers. So in conclusion, I hope that you leave the talk remembering that anyone with lungs can get lung cancer, and increasingly this is very important because we see this rising incidence of lung cancer in never smokers, and yet diagnosis in these individuals continues to be very difficult and often delayed because it's not well known that a young person my age might come in with a cough and actually have lung cancer. The differing mutational signatures in smokers and never smokers point to a different underlying etiology in these 2 groups. And a future direction that I didn't have time to talk about too much was that the etiology of lung cancer in younger versus older patients may also differ. And what we're seeing in that respect is that we don't see chromosomal translocations that are kind of a hallmark of lung cancer in younger people. We're not seeing those in postmenopausal women. And so we think there may be these kind of different etiology by age of presentation as well. So with that, I would just like to acknowledge my awesome lab. This is clearly pre-pandemic here, and CETA has done the majority of the work that I talked about today in collaboration with Dr. Garnet Anderson Amy Pigurigan, and Gavin Ha. I'd also like to thank the patients and their families that participated in the Women's Health Initiative, as well as our funding support from many different foundations, but including most importantly, the Prevent Cancer Foundation, which has allowed us to move into this new direction of cancer prevention that I'm really excited about. So thanks for your attention. I look forward to the panel discussion.